Hello,
Hello,
how are you?
just you and me, huh?
Yeah,
Okay, yeah, I'm doing fine.
good.
I'm using Mathematica now with they call Notebook Assistant,
What's that?
which is an AI. Well, I rated ChatGTP as being like a toddler.
Right.
The Notebook Assistant is like a 9-year-old.
Okay.
Not terribly smart, but getting there.
Yeah. Well, that's good.
And it tells me things which the staff says are not true.
Okay. Yeah.
Okay, so anyway,
Yeah.
at least it's faster than trying to find an expert on Mathematica and ask them questions and send them your code.
Oh, that's true. Yeah. should be able to be helpful there, I guess.
It doesn't complain. It doesn't check the time.
That's great.
Okay, I guess you can go ahead. We got two people.
Yeah, we got some people.
Soham.
Shubhaam and Shubhaam. So, hello. How are you guys?
Hi, I'm good. How are you?
All right. Great.
Fine, sir. How are you, sir?
How are you? Could you introduce yourselves?
So, I am Shubham from India, and I'm currently doing my undergraduate from Chandigarh University, Chandigarh. And I am a computer science student and looking forward to work with you, sir.
Good. Yeah. Welcome. Yeah, that's good. Soham?
Yes,
Yeah.
sir.
Hi. Oh, did you mean Soham or Shubham?
What was that?
Did you mean Soham or Shubham? We have very similar names.
Yeah, just introduce yourself, I guess.
Oh, yeah. I'm sorry. I'm unable to access my webcam. So I am pursuing my undergraduate at IIT Madras. I came across the research work on C. elegans, so that piqued my interest. So I was just hoping if I could contribute something in that direction.
Okay. Yeah, that's great. Well, thank you for attending and introducing yourselves. We do these meetings once a week, and we cover a number of topics. So, you don't understand something, that's okay. You're supposed to just soak in what you can't understand, and if you're something you really want to cover, or if you want to present something, you're welcome to do that. Or if there's something you're really interested in, please ask questions or whatever. So that's good. I assume you both have been through the OpenWorm Slack. So that's where, we do some posting there, like different discussions and things. And then the Google Summer of Code activity will start up kind of in the early spring, where we'll have some projects. I think we'll probably continue on with the projects that we worked on last year. So that description will be available soon. And if you have questions about that, that's something you can ask as well, because we don't really have a maintainer for that repository. We just have the repository there. It's in the DevoWorm GitHub, and it's DevoGraph. So that's where that repository is. And again, it's not active all year round usually. We usually do a lot of development in the summers, and then rest of the year we have sporadic engagement with it. But we'll be following up on those projects, and I'll kind of explain what those are later. Looks like Susan's here. Hello. So how are things for you this year? Did you get your thesis submitted?
Yeah. On Friday, I uploaded my thesis to MSpace.
Oh, good.
That means I added it in.
Yeah.
So, yeah, different than having to make 2 copies of it, one in color and one in black and white, and drag it over to the office.
Yeah.
It's the last time. And I came in by, what was it, 4 o'clock? And I got there at 5 to 4.
Oh, wow.
And then there were a couple people behind me, and I said, "Oh, I'm not the last one." Anyways, this time, I hope it was okay. I handed it in at 6:30 in the evening.
Oh, good. Yeah.
And if it isn't, they can give me an extension because it's there. I don't want anything more to do with it. I'm kind of annoyed at it. I didn't get all my permissions, so I had to change 4 graphs and 2 diagrams.
Oh.
Yes, that I had to make myself all on Friday.
Oh, yeah.
So it's like, I'll probably get the permissions late this week.
Oh, yeah.
I don't know.
So, yeah.
So you better.
So everything is okay then. Like all the.
Yeah, I think so. Yeah, reasonably. And if they say, "Well, we're sorry, you're late handing it in. You can't graduate till June," I'll go, "So what's your point? I don't care." Anyway, yeah, I'm kind of done with it. But looking forward to new ones.
Yeah. Well, that's good. Yeah. Congratulations on that.
Okay, some salamander stuff, and then Dick had another tensegrity idea, so
Oh, yeah.
lots of things to do.
Yeah. Yeah.
Maybe fewer deadlines.
Right. Yeah.
Like, I'm just enjoyed this weekend because there were no assignments due or no deadlines that I had to meet or anything like that. I'm like, it's just like summer vacation. Yeah.
So you said there was further ideation about the tensegrity. So where do you see that work going?
Well, for one thing, I was invited to write a paper with Dr. Zhang on the tensegrity thesis. Maybe just a 10-page thing saying what I did. And then I enrolled in APS March meeting to present it for 10 minutes. I still have to run that by the 2 professors. I can drop out if they yell.
Yeah.
Anyway, and then what was that? The nucleus. Yes, the balancing of nuclear forces are probably a tensegrity, Dick says. So.
Oh, right. I heard about this.
Yes, I don't think it has anything to do with DevoWorm exactly, but
No. The worm has atoms.
Yeah, everything has atoms.
But yeah, not directly dealing with them.
Right. And what was the other? Oh, yes. Like Dr. Sharif has said, yes, I can use the equipment when it's not being used, and I can get some images of salamander eggs developing of their blastocyst, because you can't see individual cells, but you can certainly see the blastocyst developing, and that's all I need. Like, I just wanted to tell biologists out there that this technology is available, and my main motive is to have the biologists lining up at Dr. Sharif's door. Several layers deep. Like, I would, Revenge of the Nerds. It's silly that it isn't being used, because you can image a salamander egg and have it grow up and carry on. So
that's good. Yeah.
Yeah. I showed you my PowerPoint about it, but I think I'll make a short PowerPoint for Dr. Sharif. It's not as detailed.
Yeah.
To sort of introduce him to the concept and say any other method that has been tried to image blastoceles has basically destroys the egg.
Yeah.
Yeah, anything else like MRI, X-rays, of course, cutting it up, like it all destroys the eggs. So
Yeah.
anyway, so that's kind of what I'm doing, I guess.
That sounds good. Yeah. Yeah. Good luck with the work on tensegrities. Sure, there are other ideas. Like, I don't know, do you see yourself like continuing on with the, like in general, like continuing on with the biotensegrity work?
Yeah, well, I wanted to make the 7-cell tensegrity that kind of fits. There's one in the middle and then surrounding it. I would like to try to make that, and I couldn't get my hexagons to line up. So Dr. Zhang says, no, just do the simple one. I mean, it is a subset of it, and so I've done that, and there are more things I could do with the simple one, but I prefer to do it with the 7-cell one because it's more tissue-like, obviously. So I'll make one.
Yeah.
And then I would like to find a more open source finite element program to use, or one that could be manipulated or something, but I really don't want to have to program in Linux to do it. So I'm not sure. Why am I resistant to programming in Linux?
I don't know, but a lot of times people use like software for their thesis, like say like COMSOL, and then they have they don't want to ever experience that again. So they
Well, I know how to manipulate it, sort of. But if I was to do a proper job of it,
Yeah.
I would be rewriting the software for COMSOL.
Yeah.
Yeah, because
Maybe they'll hire you.
I don't want to be hired by them. No.
Well, I had one student who went to work for them.
Oh, okay. Well, it's a finite element program, finite element analysis. And from what I can see, it is not compatible with tensegrities. Because you can build, there's a famous tensegrity that's an X, and then it has strings on the top, and then there's another X on top of that. And if you build that in COMSOL, it just falls down. It just does not compute, does not work. That's because it's too bouncy. Like if it changes shape a lot, then it figures the bridge fell down.
Oh, okay.
Yeah, and it's not a bridge, it's a tensegrity, and it will change shape. That's what they do. Anyway, it's just the reason why I got away with it was because I built the most stable tensegrity I could find.
Yeah.
It lined up with the structure of cells. It was a very simple and the most stable one I could find. It's called a T4. You know what? I don't know if you know what a T4 is. But
yeah, you did talk about it before.
Yeah, I was talking about it. But the simplest 3D tensegrity is three lines together like this, with strings top and bottom and sides, and you put two of them together, and that's a T4. And for some reason, that is a very stable tensegrity. Anyway, so that's what I used, basically, for the whole thing. Yeah. Anyway.
Yeah. Well, thanks. Yeah, for the update on that. like Morgan.
Yeah, I passed. Anyway.
All right. Yeah. Hello, Morgan. So yeah, today.
Good morning.
Oh, good morning. Yeah. Today we're gonna talk about a couple papers. The first one, this is something that Dick found in the literature towards the end of last year. And this is on, I guess it's quantitative perturbation phenotype maps reveal nonlinear responses underlying robustness of part-dependent asymmetric cell division. So this is cell division, sort of like building maps and kind of looking at these part-dependent cell division events. This is in C. elegans. We'll get into that in a minute. There's this other paper here. I don't know if we'll get into it today, but this is also an interesting one. This is from eLife, the geometric basis of epithelial convergent extension. So this is not like tensegrity, but this is actually talking about some ways to analyze some of the geometries that result from some of these epithelial cell movements. So this is a cell movement convergent extension during development. And they're actually using, what are they? Let's see. I can actually look at eLife assessment to give the significance here. This important study analyzes in the original way how tension pattern dynamics can reveal the contribution of active versus passive intercalation forces during tissue elongation. The authors develop a compelling, elegant analytical framework, and this is called isogonal tension decomposition, to disentangle the passive adjacent tissues pulling and active local tension anisotropy contributions to intercalation events. This allows the generation of global maps of tissue mechanics that can help understand this process. So that basically they're looking at this, they have this analytical framework, isogonal tension decomposition, and there are two components, the passive component and the active component. The passive component are where you have adjacent tissues pulling on this structure, and then the active are these local tension anisotropies. So this is kind of going through what they're doing here. Like I said, I don't think they're using any tensegrity stuff directly, but they're kind of developing this technique for looking at this. And this is done, I think, in Drosophila. So they're using Drosophila embryo to get at some of these issues here, where they actually have, they're looking at gastrulation and then convergent extension of the lateral ectoderm and the flow of tissue from the ventral onto the dorsal side of the embryo. Kind of going through this process. They talk about a tissue tectonics analysis, which is kind of where they're looking at changes in the tissue. And then they say, well, the above tissue tectonics analysis reveals the cell scale kinetics during GBE, which is up here, which is this germ band extension process. It does not address the fundamental question of the underlying driving forces. And so this is where, intercalations are associated with localized activity of the force-generating protein non-muscle myosin 2, or they want to build a model of these underlying driving forces. So they do this light sheet imaging and segmentation. In A, there's this process where they're kind of picking out the cells, they're modeling the cells through this segmentation of cells and then putting it into this model. And then B is where they model this cell-shaped deformation. They're modeling, a group of 4 cells, and they're modeling the deformation. And then they're modeling this intercalation process here in B. And in C, they're looking at these different regions and kind of how these, how these cells deform sort of in these over time in these local windows. D is, let's see, convergence and extension of the lateral ectoderm. So this is the fold change over time. So this is where you have 50 minutes of time. You have this fold change. It's kind of from 0 to a half fold to 1 fold to 2 fold. And then you have the length, which is in red, and the width, which is in blue. The width being dorsal ventral, and the length being anterior posterior. And you get these different phases here where you have, so the GB is germ band extension, which is after 20 minutes. VFI is ventral furrow evagination, which is from 10 to 20 minutes. So you see it captures these processes. For VFI, you basically have the length has a slightly higher fold change relative to the width, and then for GBE, it diverges so that the forces on the width are changing much slower than the forces on the length. This is during germ band extension. So they have these two components of that change, the fast and the slow. So for the length from anterior to posterior, you have this germ band extension process where the fold change is increasing faster initially and then slower later. So they're able to actually take that and actually characterize it in terms of this graph here in D. So again, D is this convergence and extension of the lateral ectoderm, X fold change defined relative to the minimum length and maximum width. So they have that. E being the interface collapse rate. So the interface collapse rate is shown here. Let's see if, so they talk about the rate of interface collapses serves as a measure for the cell intercalation rate. During VFI, there are few intercalations. During GBE, a majority of intercalations are T1 transitions, while rosettes, rearrangements involving more than 4 cells, contributed significantly less to tissue deformation. So at 40 minutes, there's a noticeable drop in the T1 rate, making the transition to the slow phase of GBE. And so that's what they're doing here. They're just looking at the interface collapse. And they don't really say what the yellow and blue lines are, but they show this. So they show these rosettes, which are these motifs that they show in B and C, and then here where they have a 4-fold and a 5-fold rosette. So it's like these junctions that we've been talking about with, you see like a 5 junction here for this rosette, and then you see this is the E prime is the counts, and so okay, the yellow is T1s and the blue are the rosettes, and so the rosettes are down here, the T1 is here, and then the T1 being in yellow, and then the rosettes in the blue. So you can see that the interface collapse is greater for the T1s. They're more T1s. So that's some of the modeling they've done here. They also show some things with having to do with the interface. So they have this group of 4 cells here. They have this model of the cells and the actin here, and then these relative tensions relative to the angle of contact here. And they show this in, I guess this is in culture, and then how the model works. And then they have some, they characterize this as some graphs, relative tension, interface length, time to collapse versus relative tension. And then some of these, some of these processes in terms of how the angle of contact deforms. So you have active contraction, passive extension, passive contraction, and passive extension. And those are just changes in how these cells sort of their angle of contact and how they're, how the actin and myosin on the boundaries are changing and deforming. So, I mean, I don't know how much more they have. They've done a lot of work on this paper. But I think I'll leave that to, in the interest of getting the next paper, I think I'll leave that to if you're interested in reading more, diving into the paper a little bit more. So any questions, and then we'll move on to the other paper I mentioned.
I think I might have the paper already.
Okay. Yeah, you may have sent it to me too.
Oh, I don't remember.
Okay, but that's interesting. It's that's in Drosophila, so it's not C. elegans, but it's also not mammals. But we do have these like processes that you can model a convergent extension, things that are common to different types of tissue formation. So it's interesting work. I mean, especially in light of the tensegrities. They don't talk about tensegrities directly in the paper, but I guess it's relevant to describing some of the stability. And some of their measures are, I don't know how good or I mean, I guess they're simple measures, but there are probably other ways you can measure some of these processes going on. Okay.
Oddly, Wayne Brodland took a paper that I did a long time ago and made it into a method of looking at epithelia to get the forces between cells.
Okay.
I don't know if they mentioned that or not. But, the question I've got is whether or not it can bring out the passive and active forces.
Yeah.
I'll see if I can find it.
All right.
Yeah.
Good. Yeah, I don't think they talk about that directly. I don't know how they're getting that.
Do they mention Brodland in the paper?
Yeah, I think they're doing a lot of things by extracting out of like images and the time series of the images. So that's how they're getting a lot of these measures here in the appendix. Up here. You don't see that citation in here.
Okay.
Brodland, yeah. But yeah, I don't know. I don't see it. All right, so let's move on to the next paper.
Okay.
Yeah.
I think I found it. I'll put it in the chat here.
Okay. Yeah. There we go. Yeah, this is 2010 Brodland and authors. This is video force microscopy reveals the mechanics of ventral furrow evagination Drosophila. So this is that system. And yeah, so this is the citation here. All right, thank you for that, Dick.
Sure.
Yeah. So this is kind of shifting gears a little bit. This is where they're talking about quantitative perturbation phenotype maps, which is, well, we'll talk about what that is. Reveal nonlinear responses underlying robustness of part-dependent asymmetric cell division. So part-dependent is a type of, it's a, we'll talk about what par is, and then the asymmetric cell divisions related to that. So, okay. So let's start with the abstract here. So this is actually published in PLOS Biology. A key challenge in the development of an organism is to maintain robust phenotypic outcomes in the face of perturbation. Yet, it is often unclear how such robust outcomes are encoded by developmental networks. So they're talking about gene expression networks or gene regulatory networks largely. So, how do you basically how does the organism correct itself when it's exposed to a perturbation? So it's like, if you expose it to a low temperature, variation in temperature, or a variation in forces, what is the effect of that? And, we know from like developmental systems that oftentimes there is this sort of robustness that happens that if you expose, sometimes there are characteristic responses to perturbations. Sometimes there's this sort of robustness that happens. So development just doesn't become incoherent. Usually you have some sort of characteristic response. I call them reaction norms or something similar. So that's what they're kind of getting at, what's going on, sort of at the developmental network level. Here we use the C. elegans zygote as a model to understand sources of developmental robustness during par polarity dependent asymmetric cell division. So par is a protein that gets expressed. You have this protein dosage that we're going to be interested in, and it's going to be, this is going to drive asymmetric cell division, which is in cases where, you have differences in size in the cells, or you only have one cell that results from a cell division. So that's what they mean by asymmetric cell divisions. Differences in the lineage tree where you have changes in the size of the daughter cells, or you only have one daughter cell, and it's not symmetrical. Usually you expect to have a mother cell give rise to two daughter cells, and then those daughter cells give rise to daughter cells. So by linking quantitatively linking alterations in protein dosage to phenotype in individual embryos, we show that spatial information in the zygote is brought out in a highly nonlinear fashion, and as a result, phenotypes are highly canalized against substantial variation in input signals. So this is where, again, we draw from Waddington's idea of canalization, where you have this sort of robustness, you have this phenotypic buffering, they call it, and depending on the input perturbation, you end up with a very similar type of phenotypic output. So in other words, if I perturb my embryo in different ways, and there are all sorts of perturbations in development. Sometimes there's developmental noise, sometimes there are variations in the environment, sometimes there are other things going on. This ensures that the process is sort of regularized, that it doesn't vary too much from embryo to embryo. And so this is this canalization process. So phenotypes are highly canalized against substantial variation in input signals. So that means if you have all these perturbations acting on the embryo, that it doesn't really have much of an effect in the overall phenotype. Our data point towards robustness of the conserved par polarity network that renders polarity axis specification resistant to variations in both the strength of the upstream symmetry breaking cues and par protein dosage. So in development, you have these symmetry breaking cues, but you also have this protein dosage dependence where you have different amounts of par in the cell, in the mother cell, and then so there's this resistance to variations in these. So this is this process that sort of smooths over a lot of what's going on here. Analogously, downstream pathways involved in cell size and fate asymmetry are robust to dosage-dependent changes in the local concentration of par proteins. So this means that we have these other pathways that are acting on, sort of as instructive cues for cell size and for differences in fate. So as the cells differentiate, we expect, say, for example, a mother cell to give rise to two daughter cells, but sometimes those daughter cells will differentiate into different cell types. Sometimes they'll acquire different fates. And so that's, this sort of asymmetry. Sometimes we have these cell size cues, so sometimes purposefully the cells will be maybe a little bit smaller, a little bit larger than others. And so this is something that is, maybe needed for development to proceed. And so what they're saying here is that these sorts of signals are robust to dosage-dependent changes in the local concentration of par protein. So if you change the concentration of this protein, the dosage, it will that those sorts of processes are robust to this signal. So it almost seems like there are these two different signals that, multiple signals that are occurring in the biology. And in embryogenesis, you have these mechanisms for robustness. You have these mechanisms that sort of override other mechanisms. And that's kind of what they're kind of trying to unpack here. So this point here about how these downstream pathways converge and they're sort of robust to these changes in protein concentration. This implies non-trivial complexity in translating par concentration profiles into pathway outputs. So there's this, your concentration of this protein par, it acts sometimes as a signal. Sometimes the developmental process is robust to changes in that signal, maybe if they're stochastic. And you have this complexity in translating the concentration of par into outputs of, daughter cells and what the daughter cells are doing. So Dick has this part outline highlighted here, this sentence. We propose that these nonlinear signal response dynamics between symmetry breaking par polarity and asymmetric division modules effectively insulate each other or insulate each individual modules from variation arising in others. So if you have these different mechanisms or different gene regulatory networks, they're all kind of acting semi-independently. They're exhibiting variation, and they're sort of insulating each other from this variation. So I guess that means that they're sort of correcting each other and maintaining this robust response in the phenotype. This decoupling helps maintain this embryo along the maintain the embryo along the correct developmental trajectory, thereby ensuring that asymmetric division is robust to perturbation. So this again is where we want to make sure that this asymmetric division, when these events are triggered, that they don't lead to more variation in the lineage tree. We also want to maintain that there are not these multiplicative effects of noise, so that when you have different gene regulatory network pathways interacting, that they're not amplifying the noise of development, and those are all kind of in the system. So,
Bradley, there's something that I'm not sure this paper addressed. It's an important question because especially in a mosaic organism like C. elegans, and that is, if you have an asymmetric cell division, and then the cells that are produced also have an asymmetric cell division, and there's an error in each one at each step, why don't the errors accumulate?
Okay, yeah.
And I'm not sure this paper addressed that, though they're beating around the bush about it.
Yeah.
Yeah.
Yeah, I guess they're just they're kind of summarizing it all as a sort of error correction mechanism, and then.
Yeah, but what is the error correction mechanism? That's what I'm getting at. Because I'd expect in an organism where most of the cells undergo an asymmetric cell division for the errors to be accumulating, and yet they don't.
Right. Yeah. Well, let's see. Yeah, let's see if they. Well, I don't know. They might talk about it in.
Okay. See, as I said, it's an important question, but I'm not sure they have answered it.
Right. Right. So they kind of talk about sort of robustness and what's going on there. The robustness of phenotypic outcomes have many origins and include mechanisms that act at multiple scales of organization. They talk about molecular buffering or dosage compensation mechanisms, and these can actually compensate for variance in network components. So, sometimes you have dosages of, say, genes or proteins that vary across, embryos. You might have a gene regulatory network where there's variation in the dosage. You might have copies of genes that arise, and they might contribute to the sort of the output of the gene regulatory network. And so, one way that gets sort of corrected for is to have this sort of variance compensation. And so they kind of talk about that, these citations 6 through 9. Network features such as activity-dependent feedback, saturation, or kinetic linkage can also ensure that input-output functions of the network remain robust to variation in particular components. So this means that you have these sort of different mechanisms, like you have feedback in a feedback loop, and it depends on what kinds of things are being expressed, what cells in the lineage tree are on. You're looking at different cells in the lineage tree. What are those, and what is the activity relative to those cells? You have saturation, where if you reach a certain sort of stoichiometry of some protein or gene, that it sort of saturates, and any more variation in that doesn't really matter. Or, I don't know, I'm not familiar with kinetic linkage, but basically there are these different mechanisms that allow the network to remain robust to variation.
Bradley,
Yeah.
a suggestion here. Two gentlemen have just joined us. Might want to consider this as a very important project that they could undertake, and that is to test models of robustness, maybe by simulating errors and how they accumulate during development, and whether or not and what mechanisms are possible for compensating for errors.
Yeah.
And I think look, the nematode has 1,000 cells. Or what is it, 800 to 1,000, depending on which kind it is. And by the time you accumulate errors, you probably have a lot of error accumulation, because that's all coming from a single cell, the egg. And yet all nematodes of a given species look alike, right? So how do they do it?
Right.
Okay, this paper, as I said, looks like it says there are feedback mechanisms and whatnot, but it doesn't quantitate them, and it doesn't show that the errors don't accumulate.
Yeah.
Okay, so modeling that might be very important, because some models, the errors will accumulate and some they won't. Now, I do have a method that perturbs the embryo so much that it can't do what it's supposed to. Put it in here. We only got from the stage of an abstract.
This is not from 1991. Defects due to compression of amphibian embryos at primary neural induction. So this is where you perturb it.
Yeah, has anyone grown a compressed nematode?
Oh, yeah.
Okay, this perturbed this particular embryo that we were dealing with to the point it didn't work.
Yeah.
And what you do is you just compress it between two sheets of glass.
Yeah.
Okay, so you can exceed the ability of the embryo to compensate, because glass is stronger than embryos.
Right.
Okay, so we didn't kill it. It just reaches the point where it stops working. So that's one way to address the question. And suppose you could even simulate that. If you can simulate the development with the error of accumulation, you could then simulate the development between two glass sheets because the cells are not allowed to go beyond the glass boundaries.
Yeah.
Okay, and so you can see what happens. And that might predict what would happen to, if no one's done the experiment, might predict the experiment.
Yeah. So yeah, there was a question, Soham, if we use the hypergraph of C. elegans embryos in some way to check error accumulations. Yeah, we could do that. I mean, the whole idea of hypergraphs, the way that we're kind of moving with them, is that hypergraphs allow us to look at different levels of complexity. So, like, we in the hypergraph models that we have like different nodes which represent maybe like they could be genetic regulatory network outputs, or they could be individual genes, or they could be cells. And then there's this hypernode which represents sort of its class. So, you might have a class of genes or a class of outputs, or even like a class of cells, or like a tissue. So we can model those at different levels. So yeah, you could actually do that sort of error accumulation checking. It would just you'd have to build the model so that you would have a way to sort of introduce error, have it represented in the node, and then see how it propagates through the network, see how it affects the state of the hypernode and things like that. Now, I know both of you have been interested in this hypergraph work since that's sort of the thing that we're going to be focusing on for the summer. But I do have a preprint you can read, and I'll put it in maybe it's already been in the DevoWorm Slack channel, but I'll re-up it so you can read more about how we approach this problem. So I'll do that. But yeah, this is something you can address with hypergraphs. What they're doing in this paper is it's different. They're dealing with actually different types of networks. They're dealing with gene regulatory networks, and they're dealing with lineage trees, which are directed graphs. So they're doing things with graphs in networks here. But yeah, you could use any different types of formulations. I think a hypergraph would be an interesting way to do that because you have this sort of multi-level analysis.
Yeah, I think this is one of the most fundamental questions in embryology. It's a little spooky because, in a sense, what you're saying, the error correction mechanisms tell the embryo what it's supposed to do.
Yeah.
And it's a little hard to believe that the individual cells know what they're supposed to do to produce the embryo.
Right.
Now, one way they might do this is by some sort of global mechanism where each cell contributes to some global mechanism, such as the electric fields that Michael Levin has been working on. And those feedback, so you get a global, local feedback that controls where the embryos, what the embryo is becoming. Okay, but I don't know if that actually works.
Yeah.
Okay, it's just a speculation at this point. So error accumulation, especially in organisms like this that are mosaic, where you expect each cell is supposed to do something, but then if it makes an error, a tiny error, why don't those errors accumulate?
Right.
Okay, it might be a global mechanism, it might be something else. So I'd really encourage that we do some research along this direction to see if we can discover how error accumulation is avoided.
Yeah.
For organisms like us, where many cells, each cell type consists of many cell types, many cells instead of just one, as in the nematode, the problem becomes even worse.
Yeah.
Okay.
Gonna say, don't forget the mechanics.
You know, and don't forget the mechanics. But you see, what got me onto this is, what was his name? Dennis Dennett, a philosopher who delved into how embryos might work. He came up with a model of trains, construction trains that build things.
Right.
Okay, not birds.
Right.
Okay, and his model was that development proceeded by one crane working on top of another. So a crane would hold a crane, would hold a crane, would hold a crane. But why doesn't the whole structure fall down? Because it'll accumulate errors. If you ever tried to, for instance, balance, well, a simple one where you build a stack of children's blocks, and you build a stack, eventually it falls over. Okay, and it doesn't take that many blocks for it to fall over, because as precisely as you want to stack the blocks, if you don't get it just right, you're off.
Right.
You all error with each one. Okay, so that's sort of a model for cumulative errors. Yeah, you can try that if you have any kids in your family.
Yeah.
Or just build it. But so it's a real mystery as to what the actual mechanism is to control error accumulation. And some good modeling might be worthwhile doing to see what works and what doesn't.
Yeah.
Okay.
All right. Yeah. So yeah, that's kind of what they don't. Yeah, I don't think they really get at that too much here. They're just looking at these mechanisms. So they talk about the C. elegans, which of course is this type of deterministic mosaic development where these lineage tree basically, you have this these binary division events, and the divisions are deterministic in the sense that we know what the cells should do when they divide. They have a nomenclature. It doesn't vary across individuals, and ultimately those cells will produce a certain tissue type or a certain cell type. And so, we have this, which is basically a deterministic system, but we do have this error that comes into play, and there's variation across cells and variation across different divisions. And so then that's what they're getting at here. So in C. elegans and related species, the first cell division is nearly always asymmetrical in both size and fate. So they're talking about that first division, which produces a two-cell embryo, the AB lineage and the P1 lineage. And of course, AB is anterior, P1 is posterior, and there's this asymmetry that gets established there. And so then that's where the first sort of asymmetry emerges, and then you get these other asymmetries or these asynchronies between cell divisions that produce, this sort of symmetry breaking, so that you get differences not only in different sublineages but also like between divisions, and those accumulate in a way that's not error. It just accumulates in terms of structure, but you can have these errors that accumulate as well. Um, so basically, you end up with this divergence in distinct lineages. Although the precise magnitude of these asymmetries can vary between species, both cell size asymmetry and cell size asynchrony are highly reproducible within a given species. So this is the case in C. elegans, but not in, say, like mammals, where you have different sets of mechanisms at play there. Moreover, at least in C. elegans, asymmetric division is robust to genetic and environmental perturbations, including both temperature variation and physical deformation. So we talked about those where you can vary the temperature in development, where the embryo is being reared in like maybe 15 degrees Celsius versus 25 degrees Celsius, or where you're exposing it to physical deformation of some type. Well, embryos can tolerate some variation in cell size, asymmetry, and cell cycle asynchrony, in part due to compensatory behaviors that occur later in development. You have these citations 34 through 36. The design principles, which is, I would go with that phrasing, that underlie this robustness of division asymmetry itself remain largely unknown. So they don't really talk about it. They just want to get into the mechanistic study and, kind of talking about these PAR proteins, which are basically partitioning defective proteins. So these are cell polarity proteins that accumulate in cells, in the mother cells. The PAR proteins consist of two antagonistic groups of membrane-associated proteins that segregate into opposing anterior and posterior membrane domains during the first division. So what happens is you have the one cell embryo, and you have these PAR proteins that are produced, and they're membrane associated proteins. They move to each end of that cell, the anterior end and the posterior end. The cell divides. Segregation is triggered by a set of semi-redundant symmetry breaking cues. So you get these PAR proteins that move to opposite ends of the cell. You get this process of segregation that's triggered by the symmetry breaking cues. And then you get this, it sort of depends on what the distribution of PAR proteins are. So there's an initial distribution that contributes to the diffusion of these proteins to different ends. And then the asymmetries are then reinforced and maintained through a core set of feedback interactions to generate robustly segregated and posterior PAR domains. Once formed, these PAR domains direct the spatial organization of downstream processes that orchestrate the size and fate asymmetry of cell division. So these PAR proteins are fundamental in sort of establishing polarity and asymmetry in the lineage tree. So you have, this sort of initial division into two cells, and then these PAR proteins continue to sort of set the pace for that asymmetry throughout the lineage tree. And so then in this study, they talk about sort of the way that PAR proteins are regulated. So there are a set of genes that regulate PAR protein production and segregation in the cell. There are 4 genes that restrict membrane association, anterior PAR proteins that are encoded by these 4 genes here. APARs, PAR3, PAR6, PKC3, and CDC42. Then you have also these PPAR, which are the posterior PAR proteins, PAR1, PAR2, LGL1, and CHIN1. So these 4 genes on each side, they sort of produce anterior PARs and posterior PARs. And so, there is this sort of these genes that code for these different isoforms of PAR, and then they end up segregating in a way that's functional. Then basically the inhibition of these genes can actually affect this feedback. So the feedback is where you have a certain number of genes expressed in the APAR category versus the PPAR category. And I guess there has to be a balance of those two. And when there isn't, or when there's, in coming to that balance, you get this positive and negative feedback. And it's reciprocal because you have basically a competition between the APARs and PPARs. So that's basically, it's what they call mutually antagonistic feedback. And so PAR polarity generally relies on balancing APARs and PPARs, which act via their respective polarity kinases, PKC3 and PAR1. And so people have mathematically modeled this sort of mutual antagonism, and this predicts sensitivity to dosage changes, which manifests as shifts in the membrane concentrations change, relative domain sizes, and ultimate collapse of polarity depending on the magnitude of the perturbation. So to recap, these different sorts of, polarity is controlled by these different classes of protein, or actually these different classes of protein their genes that are encoding these proteins and their abundance, you get this antagonistic feedback where you have this sort of balance between A pars and P pars. That controls different this mutual antagonism kind of controls different processes, these regulatory processes. And if you perturb the embryo, it can usually polarity is robust to these kind of perturbations, but sometimes it can collapse based on the magnitude of a perturbation. So oftentimes, if you perturb the cell, in a very light way, if the perturbation is weak or moderate, the embryo can overcome that. But if you have, like Dick said, this massive or catastrophic perturbation, it can't recover. So basically, you have this process polarization by cues. So there are these cues that allow for these different par complexes to separate out. Then you have this antagonistic feedback. So A par versus P par feedback specifies the par domains. Pars instruct spindle position, fate asymmetry. So basically, the pars at each end are specifying how the cell divides. So you see that when cells divide, you have this spindle. These spindles have to be in a certain position. They result in an asymmetry or an asymmetry. And then once the cells divide, the daughters are asymmetric in size and fate. That's basically the model here. And then you have, of course, the genotype where you have these genes, the A pars and the P pars. There are 4 genes for each. You have the protein dosage, which results from the gene expression, or the abundance, or the level of gene expression results in protein dosage, which results in protein abundance. This gives us polarity. And then when we get a number of rounds of this polarity mechanism, we get size and fate asymmetry. And then that can lead to differences in viability. So if we have too much asymmetry or not enough asymmetry in the embryo, we can have inviable adults or inviable worms. Basically, they don't develop correctly and they don't survive. So that's kind of how this genetic mechanism can lead to sort of this organismal effect of fitness. And then, of course, they talk about robustness mechanisms in C, where you're balancing out these different networks, these different mechanisms, and a perturbation that gets amplified in one is corrected by another one where things are stable. And they don't get into that too much, but there's a sort of a systems question here that's important. The compensation, of course, and the network properties. And this is where we get into a lot of the network questions we were talking about. There's a lot of literature on robustness and a lot of things having to do where people have kind of approached the question of robustness in biology from a network standpoint or from a complex system standpoint. And we might go over some of that work later just to get a sense of how people are thinking about that. But I think, yeah, there's definitely a link between sort of this robustness and then how different networks represent that robustness and how the network topology actually plays a role in conferring robustness on a system. So there's a lot of jargon here in terms of how they're thinking about these mechanisms, but I promise you there is sort of a way forward here. So yeah, they do a lot of mechanistic work in this paper, and I don't know if they come up with any strong conclusions in the discussion here. It looks like they have methods. So if we go back up to discussion here, they talk about robustness, heterozygosity, and nonlinear dose response curves, kind of talking about some of the genetics of this and kind of how overexpression can, there is this aspect of being robust to overexpression and variations in gene expression, which then, of course, leads to the assembly of these par complexes and their segregation in the cells and how that plays a role in conferring these these sorts of asymmetries. So they kind of get into some of this here. Then they say, what do we mean when we say that the system is robust? So they get, this is very sort of molecular, in the molecular sense of how it's robust. But basically, it confers the system sort of this sort of resistance to being vulnerable to stochastic changes in the environment, these sort of fluctuations in the environment, both molecular and out in the external environment. And yeah, they talk about critical versus subcritical perturbations. So not all perturbations are equal. Sometimes there are perturbations that can overcome these robustness mechanisms and result in a catastrophic failure of the system. So this is something that's really interesting as well, not just that you can, that you have this sort of set of robustness mechanisms, but there is this sort of nonlinearity where you have these, robustness, that robustness only works up to a certain point. And then you can have a catastrophic perturbation that collapses the system or renders it inviable. Yeah, so they get into the, so yeah, I don't think they really talk about the theory too much or about some of the computational modeling. They talk a little bit about mathematical modeling, but not a lot. Okay. So I think that's enough for today. Looks like Dick had to go, but again, this is something for further research, and I think we'd like to maybe follow up on this paper in the new year, especially some of these mechanisms that we've talked about. Okay, any questions or comments before we go?
It's the first day of the school year, but yeah, very interesting, and Happy New Year, everyone.
Yeah, Happy New Year. Thanks.
Happy New Year.
Yeah. All right. Thanks for attending. See you next week.
Bye.
Bye.
Take care.
Take care.
Thank you, sir.